Welcome, this is experiment 4. I wanted to make a video for you. I'm just going to tell you in advance that this experiment is going to take some time. In fact, you might want to make a goal for yourself. There's 4 parts to this experiment. Or you could say there are 4 experiments within the larger lab assignment. And I probably would encourage you to just maybe do one a night if that's possible, or just set aside some time to knock this out. So I'm going to go to week 3, which is what this is under. This is week 3 lab. And I'm going to go down here. Let's see, objection. Okay, so there's lab number 3. And here's the diffusion osmosis experiment. So I'm going to go ahead, and open up. This is the answer sheet for this. And then I'll open up the actual experiment itself. Here's the rubric. Now, I've decided not to include any photos. First time we're doing this lab kit at home. In the future, students will have to provide photos for this. I've decided not to do that. I'm just going to grade you based on your data and how you answer the questions. However, experiment number 4, which I'm going to count as a bonus, it will actually count 15 points toward this particular lab report if you do it, because you have to go buy potatoes. That one you need a minimum of 5 photos. So I'll get to that at the end. But here are some lab questions, and then here are the actual data sheets. So we're going to come back to this. All right, I'm going to go here. I'm going to open up the Osmosis Diffusion lab experiment. I'm going to click on the new window, and here we go. And I'm really impressed. I really like, I know sometimes you're not required to read a lot at times, and sometimes you're going to have to read and be able to, pull out information for what you've read. So this is not too bad. It's all pretty short. It does tell you right here how to do a serial dilution. I'm going to give you a hint for that later. But what I want you to do is go through, read all this, answer the practice questions. Which the following does not impact membrane permeability. I'm going to let you answer all of these. Tonicity. And what we're doing here, this is going to test, osmosis is based on movement of water molecules. And just remember this, water always goes where salt is, or solids. And that's what you're going to be doing in these experiments. You would actually do these very experiments in a lab setting. We do these at Howard College in the first part of the A&P lab. So hypertonic solutions, those are solutions with high concentrations of solids. They have a tendency to, if you put cells or anything inside there, like a human cell for instance, they have a tendency to shrink cells. Hypotonic solutions have very little solids or no solids. They're almost 100% fluid or water. They have a tendency to cause cells to shrink because, like I said, water is always drawn where there's more solids. In that case, there's more solids inside the cell. ISO means same. So you're going to have the same concentration from the cell to the solution it's sitting in. So based on all that, and there's actually some really cool embedded in here. Maybe it's here. It is. Great virtual learning. So go through that, look through this whole thing. Now, I always encourage you to do the lab drill. And then when it comes down here to assignments and procedures, you're going to do the first 3 for a grade and do what is on your answer sheet, answer what is required. And then the 4th here will be for a bonus. It's worth an extra 15 points on your lab report. So here's the first one. The things you're going to need from your lab kit, you have some corn syrup, some red dye, blue dye. This one's pretty easy. Petri dish. And here is a great demo. I've decided to let you use that demo. I think it's probably better than what I could do at this point. So watch this first. It'll show you how to set it up. And basically what you're going to do, corn syrup. It's got a little bit of viscosity to it. You're going to pour that in a Petri dish. You're going to have a ruler to measure movement. And if you notice up here, Red dye has a molecular weight of 496 grams per mole. That's a chemistry measurement. Blue dye has a molecular weight of 793 grams per mole. So this is actually a bigger molecule. It's got more mass to it. So you're going to measure, you're going to test to see, does the size or weight of molecules have anything to do with diffusion? So it's kind of a cool experiment. You're going to fill, like it says there, fill half the petri dish with corn syrup until the surface is covered. That's pretty easy. Place a single drop of blue dye in the middle of the corn syrup. Note the position. Record the diameter of the dye every 10 seconds for a total of 2 minutes. Record your data in table 1. Repeat the procedure for red dye, the red dye. So theoretically, you're going to see, okay, does molecule size have anything to do with diffusion? And you get to find that out and enter it in your data table. I'm going to hypothesize the smaller one's going to move further. We'll see if I'm right or not. So that's your first experiment. Now, when you're done with that, so you're going to come over here to your lab answer sheet, answer the pre-lab questions that's embedded in the part I'm telling you to read, and then here's your table. See, blue dye, red dye. You're just going to enter, there's 120 seconds, so 2 minutes. It says the rate of diffusion, so you're going to enter the distance traveled. Okay? And then, I'm sorry. Yeah, the rate, sorry. And then here, yeah, the speed of diffusion. You time it. All right, and there's some post-lab questions based on that. And then we're going on to experiment number 2. This one's got some detail to it. So I'm going to go back, click on this, concentration gradients and membrane permeability. Human cells are called selectively permeable. Some certain substances can cross over a cell membrane, some cannot. That's referred to as permeability. It might have to do with the size of the molecule, the actual, what the composition of the membrane is, all kinds of things. You're going to be using dialysis tubing in this experiment. Down here in the corner, now it's got the little blue stars because you want to be careful with it. Measure exactly what you're needed because you're going to need some for later. And dialysis tubing, what you do is you soak it, and it's like a paper little plastic bag at the supermarket. You know, when you go through Walmart, that I'm thinking of, and they're stuck together and you kind of have to pry open the bags to put your stuff in. You're going to have to pry open the end of a dialysis tubing and basically make a bag out of it. Now, I'm going to post this on your site, and I believe I saved it here. I meant to post this already. But right here, you're going to be making one of these. This is called a dialysis bag. You would do this in a face-to-face lab. You're going to have both ends tied off, and you are given rubber bands for this. They're actually colored, so you'll know which bag you're placing in the right beakers. But you're going to soak these. It's actually stuck together, and on the end, you're going to need to rub it between your fingers after soaking it or getting it wet, and it's going to pop open on the end, and then you tie off the ends. And now you're going to tie off the bottom right away, and you're going to leave the top open. And I believe in this experiment, it's asking you to actually leave the top open. But you have to have one end open to fill with fluid. I will post that underneath or where this video. You'll see it. You'll see it before you click on the video. So going back, so here are all the things you're going to need. It's giving you a little breakdown here of everything. Some potassium iodine that's used to test for starch, and some 4 rubber bands. And yeah, you have to provide some water and some scissors. And that's not too bad. So you're going to test for the movement of glucose and starch. I'll just tell you, glucose is a monosaccharide. It's really small. Starch is a polysaccharide, much larger molecule. And the effect of selective permeability on diffusion of these molecules. Like I said, selectively permeable means certain things can pass through and certain things cannot. Once again, this is sort of based on, say, glucose is pretty small, starch is big. Read through this. It says, do not allow the open end of the dialysis tube to fall into the beaker. This is why you're doing your experiment. And there's a picture provided for that. So here's what you're going to do. Measure 50 mils of water into a 100 mil beaker using the 100 mil graduated cylinder. Here's a nice little video to show you how to do that. And it also shows you how to make shows you about the dialysis tubing. Cut a piece of dialysis tubing 15 centimeters long, submerge it in water for at least 10 minutes. This is so you can try to get the end open. You're going to tie one end with a rubber band, and the other end you want to get it open because it's actually two membranes stuck together. It's a bag once you tie the bottom off, so you can fill it with fluid. Okay, measure 82 mils of water into a second mil beaker using the 100 mil graduated cylinder. That's pretty easy. Label this beaker experimental with a permanent marker. This is where you're going to put the filled dialysis bag in for step 10. Now you're going to have to make a glucose, it says / sucrose solution. Sucrose is actually a disaccharide, so it's a little bit bigger sugar. A polysaccharide or starch is not a sugar. So you're going to put 5 mils of glucose into a 100 mil beaker, label it the GS solution. Get a different pipette, those are in your kit. Remember, each pipette, actually the top line is 1 mil. So you have to do this 5 times. Add 5 mils of starch solution to the same beaker, and then you're going to mix it around. So mix the starch together along with the glucose / sucrose mixture. And what you're ending up here, you have a monosaccharide and glucose, you have a disaccharide and sucrose, and then you're going to have a polysaccharide and starch. All different sizes. I always love when that happens. So, using the same pipette you used to mix it together, remove 2 mils of the glucose starch solution and place it in a clean 100 mil beaker. Label this positive control. Now you have some test strips in your kit. You want to make sure you can test to see what a positive test looks like, because you're going to test to see during your experiment, did glucose move out of the bag or did starch move out of the bag? And you're only going to know, you have to know what a positive test looks like. So you can test for the presence of that during your experiment. Otherwise, you're not going to know. You can do your experiment. Did the molecules move? I have no clue. Well, this is why you're doing that. So once you get to see what a positive test looks like, then you can move on from that. Now it says, using a clean pipette, remove 2 mils of water from the experimental beaker and place it in a clean 100 mil beaker. Label this one negative control. So in other words, this should not have any glucose in it. So that way you can see what a negative looks like. So you're going to be testing, did it move or not move? If you test and it's negative, then you know no movement occurred. If it's positive, then movement did occur. And what moved? Did the glucose move? Did the starch move? So that's why you do a positive and negative control, so that you can test for the results during your experiment. So you know what a positive looks like and you know what a negative looks like. Okay, after 10 minutes have passed, remove the dialysis tube. Close one end by folding over 3 centimeters on the bottom. And I think what I'm going to do, I know YouTube has, I've made these a ton. I'll just put a YouTube video probably right below this video, and it'll show you how to make the bag if you want. So draw off the sides, add a small amount of water. Okay, that's testing to see if it leaks or not. I don't know if that's really necessary. If you tie off the end and put a rubber band, it should be sealed. But okay, using the same pipette you used to mix, you want to add 8 mils of glucose starch solution into the prepared dialysis bag. One end's going to be tied off. The other end you want to pop open. You want to rub it between your fingers and get one little end of it to pop open, and then you can add your 8 mils. So place it in the experimental beaker as so. Allow it to sit for 60 minutes. That's exciting. And if you, well, anyway, that's an hour. So It says clean up the, remove the clean and dry all materials except the beaker holding the bag. So after it's diffused for 60 minutes, remove the dialysis bag from the beaker and empty the contents of the bag into a dry, clean beaker. Now that's the one you're going to use to test. Okay, you're going to see, did glucose move out? This thing is irritating. Did glucose move out? Did starch move out? And that's when you're going to test to see if it's positive or negative. You're going to add this for starch, this potassium iodide, IKI, that's the test for starch. And it says using a pipette, transfer 8 mils of water into the experimental beaker to a clean beaker. Test that beaker water for the presence of glucose by dipping 1 glucose test strip. That's how you're going to test for glucose. You test for starch by adding this, and then record the final color into the table provided. So we're going to go back. This is table 2. Here's your first table. It says the indicator reagent data, your dialysis bag after an hour, beaker water after an hour, and then here are your post-lab questions. What I want you to do is put thought into these, answer the questions. Sometimes there's not a right or wrong answer. And if you do your experiment correctly, then based on the data, and I have the numbers for that, and what should have moved and not moved. Okay? All right. Now we're on to the last ones that count, which is experiment 3. I'm going to close this, go back to this. Same thing, kind of have a few. Now you're going to have some beakers here. You got 3 2-mil beakers for waste beakers. This is a very common experiment that we do. I have to give you some hints for this one. So you're going to make 4 dialysis bags. Yeah, just like you did. Submerge them for 10 minutes. After 10 minutes, remove the beaker. Do exactly what you did before. So this will open the dialysis tubing. It says use your thumb and index finger to rub the dialysis tubing between your fingers. This will pop open the end. Wrap one of the yellow rubber bands around one of the dialysis tubing. Leave a few inches past that. Make sure it doesn't leak. And then repeat the steps for the remaining dialysis tube and the 3 remaining band colors. I believe you have yellow, blue, red, and green. Okay, I believe. You have this in your kit. Reconstitute the sucrose powder. You're going to add 160 mils of warm water to give you 200 mils of what's called 30% stock sucrose solution. So on your table here, when it says, well, actually, let me go back. Here, that's what you're going to use when you're making your beaker solutions. To make 30%, you take 10 mils out of this, and then you're going to add, well, you add no water. You're going to put that to a beaker. To make a 15%, you take 5 mils out of that stock solution, and then you add 5 mils of water. This is going to be your isotonic, the same solution. This is your hypertonic solution. This 3% says take 2 mils out and then add 18 mils of water. This is going to be a hypotonic solution. Now, here it kind of throws in this out of nowhere. It says use some of the remaining stock solution to create an additional 200 mils of 3% sucrose solution. What I suggest you do here, because you're going to do your mixtures here, that's 17 mils, and then you're going to add 150 mils to a 250 mil beaker, which is 167 mils of what was 200 here. So all you're going to have left is about 33 mils. I suggest you just fill it up to 200 mils with water. So add an additional 167 mils of water to make it 200 mils, and that will get you really close to 3%. Very simple. Measure and pour 10 mils of 30% from the waste beaker into the dialysis bag. So from your solutions up here, you're going to use these solutions, you're going to add them to your bags that you've made. See that says measure 10 mils of the 15% from the red rubber band, measure 3% for the blue rubber band, and then measure and pour 3 mils from the waste beaker into the bag with the green rubber band. So make sure you get those straight. See here, what they're saying is, leave this end open and you're going to tie off the ends down here. Do not let this fall in, so be real careful with that. Let the bag sit for an hour. Now, so here's the thing. Place the yellow, red, and blue band tubing in the 3% that you created sucrose solution. The one I told you to add 167 mils to make it 3%. Place the green banded tubing in the 30% sucrose, which is the one up here that's 150 mils. That's your hypertonic solution. So what you're going to do, you're trying to see, like I said, water's drawn to solids. Some of these bags are going to lose water, some are going to gain water. You get to measure that at the end. So after an hour, let them sit for an hour. So cut the tubing off, well, if you have to, or the top of the tubing may need to be cut or removed if you can't get it to pour out. That's what it's saying. And then, like it says here, you're going to measure each bag. You measure the solution volumes using the 100 mil graduated cylinder. So here's my hypothesis. The green bag is going to lose water. Okay? It's not going to have as much. I think you added 10 mils up here, I believe. Right here, green rubber band says pour 10 mils. It's going to lose some of that. These others, especially the little 3% here one, it's probably going to gain some water. So what you're going to do is you're going to measure these out, write down your numbers, and then you're going to fill out the table that is provided here. Initial volume, they were all 10 mils, and then your final volume. And then you can subtract the displacement, and then answer these questions. It's actually pretty cool. This last one here is a bonus. You do it, you do a good job with it, I'll give you 15 points. You are required to take at least 5 pictures, and you're going to put those pictures down here below these questions. So this one requires you to go out and buy a potato. I know that's not a huge expense, but sorry, in fact, let me go back. Excuse me. So this is experiment number 4. Yeah, you have to go out and get a potato. In fact, pick 2 different kinds. Russets, those big giant ones you grill and put in the oven. Get a sweet potato. Get an Idaho potato. Make sure you put that in your report, what you used. What you're going to do is you're going to cut slices of potato. What potatoes will do is they'll absorb water. And so watch this. Great demo here. It says plant cells have the ability to generate osmotic pressure. That's why when you water plants and you water the ground, plants have cell walls, so they're not supposed to burst. So they actually like hypertonic environments, or hypotonic environments, where water is being forced inside the plant cells. That's how they acquire water. So it's called tonicity. It has to do with how fluids move with solids, basically. Now it says here, using a knife, carefully cut 2 strips of the first potato on a cutting board. There's a sample 1A and a sample 1B, 10 centimeters long, 1 centimeter wide. Just kind of cut you a thin strip. It says fill a 100 ml graduated cylinder with 50 mils of water. Place the first potato strip into that, and then it says record the initial displacement in table 7. So you're gonna subtract, then you take the initial volume like it says here, and then you subtract it from the final volume. And you can read through that. Like it says there, your calculation, 7 mils. If you started out with 57 and you end up with 50, the displacement is 7. So you're going to do that with the different potatoes. So remove sample A. If any water was lost, refill it to the 50 mil mark. Place the second potato in the graduated cylinder. Record the initial displacement in Table 7. Repeat these for the second potato type. So just read through this. It says create a 20% sodium chloride solution by adding 77 mils of warm water to the 16 grams of sodium chloride. That came in your lab kit. It's in a bottle. You're going to add these to the tubes that the potato strips are submerged in. After an hour, pour the solution out from the test tubes. Record the final displacement values in Table 7. It says complete the last column of Table 7 by subtracting the initial displacement from the final displacement. And down here, there you go. Potato type, potato observations, what color, thick or thin. And it's actually a cool experiment. It's going to let you see, salt is going to affect how much water the potatoes absorb. So I'll let you do these, take pictures, with your potatoes soaking in them. I need about 5, and you're going to enter those down below your questions here. Okay? So that's experiment 4. That would actually bring you to the end of what is required for this first section of anatomy. Okay, so there's 4 experiments. They're due by October 20th. 4 lab assignments, and they're due by October 20th. So you have plenty of time for this. Okay? Good luck, and take these serious, do your best work, do your best job, and do a good job answering your questions, and I'll reward you for it.